Scientists are no less than messiahs for mankind. Throughout history, they have revolutionized the world in unimaginable ways, from making groundbreaking discoveries that made the world around us more comprehensible to healing humanity with medicine and non-invasive techniques. The genius of scientists is simply unparalleled. The human mind is beyond brilliant. Years of hard work and the genius of people generate innovations that stun the world and leave an impact engraved deep in history. Such brains, with their dexterity, expertise, and courage, go on to create global records and immortalize their names for several generations to come. A plethora of facilities that we benefit from today, whether they be related to health, technology, or travel, we owe it to these brilliant minds, their lifelong endeavors and experimentations. Tons of science experiments have led to new visionary working methods, more convenience, innumerable amenities, and just simply open doors to the impossible. One such experiment is selective breeding or crossbreeding. Introduced by Sir Robert Bakewell in the 18th century, selective breeding aims at developing species with more desirable and more robust inheritable traits. Bakewell kept records of animals' performance for objective selection. He used inbreeding, that is, mating of related animals with similar characteristics, to fix certain anomalies in animals. Bakewell succeeded in developing the new Leicester sheep from the old Lincolnshire breed. The new Leicester had good quality fleece and a fatty shoulder that was popular then. He also noticed that longhorn cattle were growing well and used less feed compared to other cattle, so he developed it further in order to grow more meat efficiently. Crossbreeding helps farmers develop more desirable cattle that fit the environment better and yield more milk or tastier meat. It is also common for different species of dogs to be crossbred, which results in much more adorable and desirable hybrid dogs. For example, the magnificent wolf dog is a cross between a wolf and a dog. A more domesticated example is the Labradoodle, whose parents are Labrador Retriever and Poodle. Cats are hybridized too. A striking example is the Bengal cat, whose wild-looking spots and marbling are owed to genes from the Asian leopard cat and multiple domestic cats. This exotic cat species, purchased from a well-reputed breeder, can cost you up to $5,000. However, crossbreeding has its disadvantages too. Sometimes the outcome of crossbreeding is a pure disaster. Things can take an ugly turn when the hybrid is not what was expected. Crossbreeding, therefore, has been deemed unethical in some cases. No matter how well-intended one might be, where do we draw the line? There have been times when scientists crossed the boundaries and carried on with their experiments regardless. Today, we're going to tell you about the 15 most terrifying crossbreeding experiments scientists don't want you to know about. Baboon heart. On October 26, 1984, pediatric cardiothoracic surgeon Leonard Bailey replaced an infant's heart with a baboon heart. Born on the 14th of October 1984, Stephanie Faye Bouclair, also known as Baby Faye, was diagnosed with hypoplastic left heart syndrome. The syndrome caused the left side of her heart to not grow properly. She weighed just 5 pounds and was 19 inches long at the time of her birth. Unfortunately, no heart donor could be found, and so the world's first cross-species transplantation was performed. Dr. Bailey had shared his research on cross-species heart transplantation at national and international scientific conferences, but to everyone's surprise, he actually went ahead with the practical procedure. His workplace, Loma Linda University Medical Center, soon became the center of attention worldwide. Animal rights advocates protested outside the university while holding placards and wearing animal skin accessories, which was a sight pretty ironic. The small city of Loma Linda was swarmed with national and international media. The ethics of the surgery were questioned too, and cross-species transplantation was examined from every angle. Dr. Bailey's motives were heavily doubted, as well as that of Baby Faye's parents. Opinions from medical ethicists were readily sought. People from every nook and corner of the world cheered on the little infant. However, as incredible as it all sounds, it did not end well. The world mourned as Baby Faye passed away just after 21 days. Human rabbit embryo. In 2003, Chinese scientists used cloning techniques to create hybrid embryos of human and rabbit cells for the first time. This experiment was strongly condemned on the basis of moral and ethical values. More than 100 of these hybrids were made by mixing human skin cells and rabbit eggs. Researchers hoped it would lead to an abundant source of embryonic stem cells for research and medical use. The experiment was conducted to know whether cells and tissues could be grown this way. Prior attempts of this type by an American scientist had failed. As per rules and regulations, the animal had to be killed once there were human cells in its brain, and the experiment was to be halted immediately. The experiment was deemed unethical by theologians. The human-rabbit hybrid was allowed to develop for a few days before it was put down. Human cells fused with pig embryos. Scientists across the globe are very keen on growing human organs artificially. As the number of human organs available for transplants is declining, they are swiftly working on growing human organs in livestock and other animals. But slowed down by critiques from bioethicists and countless ethical concerns, they haven't been too successful as of yet. In 2017, cell biologist Jun Wu and several other scientists in Juan Carlos Ibzuza Belmonte's lab at the Salk Institute for Biological Studies, San Diego, California, reportedly injected human iPS cells into pig embryos, which were then planted into sows. However, the experiment could not amount to much success, as half of the resulting fetuses were stunted and had slow growth rates. The other half that was normal had very few human cells after a month of gestation. Nonetheless, it is certainly uncomfortable to think about human cells being fused with pig embryos. One can only imagine what the outcome would have been like. If this one-of-a-kind experiment had been a success, would the offspring have looked like humans, part pig? Perhaps a mutant human pig isn't that good of an idea. Pig infused with human blood. Two scientists, Dan Gary and Mary Gary from the University of Minnesota, succeeded in developing pig embryos that have human muscle. As disturbing as it may sound, the two want to create human organs inside animals for transplantation purposes. They are working on developing pigs that have human muscle and especially human vasculature, because the recipient's body might reject the transplanted organ if it contained porcine blood vessels. Dan and Mary carried out their experiment by injecting genetically modified human iPS cells into pig embryos. After gestation in surrogate pigs for 17 to 27 days, the chimeric embryos contain traces of hematological and muscle tissues comprised completely of human cells. However, they believe such experimentations are not an efficient solution to the falling number of transplants, as the modifications could pose a major threat of cancer to the recipient. Belgian Blue. The ripped Belgian Blue is a breed of beef cattle from Belgium. The blue in its name comes from the distinct blue color it had back then. In the 1800s, farmers mixed native cattle with shorthorn instead of any other animal, and years of selective breeding resulted in this monstrous cow. The origins of how the Belgian Blue was introduced to the world trace back to 1978, when a Canadian farmer, Nick Tutt, brought with him some Belgian Blues and exhibited them in local universities in the United States. Scientists were greatly fascinated by this bulky animal. An average male Belgian Blue can weigh up to 1,200 kilograms. It can easily crush anyone to a gut-wrenching death. This breed of cattle has been created exclusively by selective breeding methods. A random genetic mutation that showed up in this species 200 years ago is what makes these cows so muscular. To keep this genetic mutation going, farmers have continued to inbreed these animals. This natural mutation inhibits the expression of a protein known as myostatin, which in turn limits muscle production to some extent. Less myostatin equals more muscle. Additionally, the same mutation seems also to inhibit fat deposition, resulting in less fat tissue. A modern version of the Belgian Blue was developed in the 1950s by Professor Hanset at an artificial insemination center in Belgium. The breed is under continuous development and improvement. Today, the Belgian Blue is not only available in the blue color, but also black, white, blue roan, and the combination of these colors as well. Belgian Blue yields meat of excellent quality, as it contains high protein and low fat. Their meat and milk are completely safe for human consumption. Despite that, there are various problems associated with the Belgian Blue. Inbreeding on a huge scale oftentimes results in abnormal offspring. Some individuals have unusually large tongues that cause difficulties in breathing and lead to suffocation and premature death. Moreover, the newborns are sizable as compared to other animals, and Belgian Blue cows have an inherently narrow birth canal. This makes childbirth extremely difficult, and cesarean sections have to be resorted to. Killer bees. How the killer bees came into being is a scene straight out of a science fiction film. Killer bees are not Mother Nature's gift, but rather a genetically modified hell unleashed accidentally. In 1956, Brazilian geneticist Dr. Warwick E. Kerr, commissioned by the Brazilian government, interbred subspecies of the Western honeybee with European honeybees and the African honeybee in an attempt of improving honey production in Brazil. Aiming to create a new species by isolating the European bee's trait of plentiful honey production and the African honeybee's adaptation to warmer climates, a less desirable trait was inherited as well. The new hybrid bees turned out to be very aggressive. This immense aggression earned them the notorious recognition as the killer bees. Dramatically enough, a handful of these hybrid bees escaped quarantine and spread through Central and South America pretty quickly. They're also called Africanized honeybees. The first permanent Africanized honeybee colonies arrived in Texas from Mexico in 1990. They are dangerous stinging insects and are known to chase people for more than a quarter of a mile once they are triggered. They are smaller than other honeybees, have shorter wings, and carry venom. When they sense their colonies are under threat, their entire swarm attacks the perpetrator. Hives consisting of 300,000 to 800,000 Africanized bees have been reported to have attacked people. A notable feature of their attacks is that a pheromone is released with each sting, which signals more bees to join the ongoing incursion. Africanized bees are quite determined. Victims are reported taking refuge underwater to avoid the stings, and as shocking as it may sound, the bees willingly wait for their prey and continue the assault once the person is out of water. The hybrid bees look like they would go to unfathomable extents to justify their name. Human mice. Like many other scientists, Dr. Hiromitsu Nakauchi from Japan aims at developing human organs in other animals. Nakauchi has conducted a wide range of biological experiments, all of which meant to develop human organs inside animals, as organ donors are rare to find for transplants. Nakauchi guides teams at the University of Tokyo and Stanford University in California. He plans to grow human cells in mouse and rat embryos and then transplant those embryos into surrogate animals. His ultimate goal is to produce animals with organs made of human cells that can eventually be transplanted into humans. In 2017, Nakauchi with his colleagues injected mouse iPS cells into the embryo of a rat that could not produce a pancreas. The rat developed a pancreas made entirely of mouse cells. Nakauchi and his team then transplanted this pancreas into a mouse that had been engineered to have diabetes. The rat-produced organ was able to control blood sugar levels, effectively curing the mouse of diabetes. In 2019, this Japanese stem cell scientist received support from the Japanese government to create animal embryos that contain human cells and transplant them into surrogate animals as the ban on this practice was lifted. In 2018, Nakauchi and his colleagues announced at the American Association for the Advancement of Science meeting in Austin, Texas, that they had fused human iPS cells with sheep embryos that had been engineered to lack a pancreas. Grown for 28 days, the hybrid embryo contained too few human cells and couldn't develop anything like organs. The evolutionary and genetic distance between humans and sheep is suspected to be the reason behind the failure of this experiment. Over the years, Dr. Nakauchi has been on the receiving end of harsh criticism for running lab experiments that some bioethicists and animal rights activists consider unethical. Smart mice. In 2013, neurologist Dr. Stephen Goldman teamed up with his wife, Micah Niedergard, to test his theory that glial cells in the human brain are far more significant than neurons. They injected some human glial cells into the brains of some mice. The mice grew up just fine with their brains comprised of both human and mouse cells. The human cells even outnumbered the mouse cells in some parts of the mouse. Goldman said that the glial cells maintained their human characteristics. The mice injected with the glial cells were reportedly smarter than their fellow mice. Although they socialized with their fellows too and behaved like a mouse is supposed to, they performed way better in the classical maze game and made fewer errors. Goldman remarked that the glial cells enhanced the learning ability of mice. Bioethicists such as Robert Streifer from the University of Wisconsin-Madison raised ethical concerns regarding this experiment and complained about how some scientists consider a mouse just an old normal mouse even after injecting them with human brain cells. Streifer further added, "It's not just that these mice can get through a maze more quickly, they're better at recognizing things that scare them. Perception of fear is one of the things bioethicists must weigh when they decide the types of experiments you can do on an animal." Goldman explained how these mice are far from becoming anything like humans, yet bioethicists from all around the world are concerned about such experiments and fear they might lead to the birth of some weird human-mouse hybrid with human-like intelligence. The Vacanti mouse. In 1997, the Vacanti mouse surfaced in a BBC documentary that doesn't look any less than God's abomination. This mouse had a human ear grown on its back. Surgeons from Harvard University, Joseph Vacanti and his brother Charles Vacanti, teamed up with MIT engineer Bob Langer to conduct an experiment that was originally aimed at a better understanding of plastic and reconstructive surgery. They implanted synthetic biodegradable polymer embedded with bovine-derived cartilage cells into the mouse. Put simply, it was an ear-shaped scaffolding made from cells of cartilage that could be grafted onto a human, or so they claimed. This experiment, however, prompted an international surge in the field of tissue engineering research and applications. Humanzee. Back in 2005, researchers sequenced the chimp genome and concluded that we share 99% of our DNA with chimpanzees. They are therefore our closest living relatives. Throughout history, many scientists have wondered whether they could generate a human-chimp hybrid or not, but a few proceeded with such bizarre experiments. The most noteworthy and historical human-chimp hybridization experiment dates back to the early 20th century. Russian scientist Ilya Ivanovich Ivanov, who emerged as one of the most controversial scientists of all time, initially proposed the Humanzee experiment in 1910 at the World Congress of Zoologists in Graz, Austria. After the Russian Revolution in 1917, things changed and the government of Russia finally approved and funded his work in 1924. Considered the real-life Frankenstein, Ivanovich was an expert in animal hybridization and artificial insemination. He had successfully conducted several peculiar experiments, such as the making of Zedonk, a zebra-donkey hybrid, and a guinea pig mouse. Despite all his efforts, his endeavors to create a human chimp in Guinea, then part of French West Africa, failed as none of the apes conceived. The Soviet scientists didn't lose hope and came up with another strange idea of fertilizing a human female with the sperm of a male chimpanzee. Surprisingly enough, 5 women agreed to volunteer for his experiment, but it couldn't proceed as the apes he brought with him from Africa failed to adapt to their surroundings and died. New apes arrived in the Russia facility in 1930, but before Ilya Ivanovich could start his experiments again, he fell victim to the widespread purge of the Soviet scientists and was exiled to Kazakhstan. Although he was released a year later, he suffered from a fatal stroke in 1932. Ivanovich died, and so did his wild dream of producing the Humanzee hybrid. Human-chimp hybridization. Attempts to create a Humanzee haven't been limited to Russia. Gordon Gallup, an American psychologist, claimed that a human-chimp hybrid was successfully bred at the Orange Park Laboratory in Florida in the 1920s. Gallup claims to have learned this information from his former university professor. The researchers who partook in this experiment had second thoughts and moral and ethical concerns about it, which can be the reason this experiment was never officially unveiled to the world. The infant that was the product of this odd experiment was euthanized, according to Gallup. No official reports of such an experiment were ever released by the relevant doctors, institutes, or the US government. However, this experiment has been cited by numerous scholars and scientists, which somewhat helps add to its authenticity. No matter whether Gallup's claims are true or it's all a made-up story, it is surely one hell of an extraordinary one, as well as a creepy case. Chimera embryos. Many latest and known experiments to produce a human-monkey hybrid have taken place in recent years. As per a 2019 report of Alpais, an international team of researchers created chimera embryos that contained both human and monkey cells. This unconventional experiment was conducted in China to avoid controversies and legal issues. The objective was to grow functional organs that could be transplanted into humans. The embryos were allowed to develop up to 10,000 cells, after which the scientists interrupted the experiment due to innumerable ethical concerns raised by scientists and ethicists. In 2021, French biologist Pierre Sabatier and his team published details of their experiment to create human-monkey chimeras. They were able to grow an embryo only made of 10 human cells and 250 monkey cells that lived for 7 days. Apart from all the ethical concerns, scientists think that with the development of chimeras, they can improve their understanding of how humans function. This will eventually help them find treatments and cures for different diseases. Chimera virus. You have probably seen films about genetically modified viral outbreaks that led to death and destruction on a global scale. Scary. In 2017, researchers from Instituto de Medicina Molecular Lisboa in Portugal created a chimera virus that allows them to study cancer-causing molecules induced by the human herpes virus infection in mice models of disease. Portuguese researchers collaborated with researchers from Harvard Medical School to create the chimera virus with a viral human gene. It apparently allows them to study viruses and how they affect rodents as well as humans. Nevertheless, such experiments raise concerns and instill genuine fears of what might happen if an outbreak does occur. Birth of human lamb. In 2017, a sheep in South Africa allegedly gave birth to an eerie lamb that resembled humans. What worsens this case is the speculation that the sheep might have fallen victim to the assault of a barbaric human. This particular incident went viral on social media and was confirmed to be real by the Eastern Cape Department of Rural Development and Agrarian Reform in South Africa. The Chief Director of Veterinary Services asked the people of that community not to panic. As per the authorities, the human lamb is not the progeny of sheep ovum and human sperm, but a deformed fetus. They insinuated that the mother sheep was infected and that viral infection was transferred into the baby lamb as well, causing it to suffer malnourishment and deformities. Authorities claimed that the so-called human lamb had a deformed head due to an abnormal accumulation of fluid in the brain cavity, a smaller brain, and an underdeveloped lower jaw that caused it to look like that. Whatever the reason lies behind the way that lamb turned out, it definitely creeped out the whole village, the farm men, and their families. Pyrenean ibex resurrected. In January 2009, scientists successfully conducted an unreal experiment of resurrecting an extinct animal. Pyrenean ibex, a subspecies of the Spanish ibex, was rapidly decreasing in number due to hunting. In 2000, the last of their kind passed away. A team of multiple researchers led by Dr. Jose Folk of Zaragoza from the Center of Food Technology and Research of Aragon used frozen skin samples of the extinct species to make clone embryos. The skin samples were taken in 1999. The clone embryos were made by injecting the DNA into goat eggs. The clones were then planted into other subspecies of Spanish ibex or goat ibex hybrids. The cloned newborn lived only for a few minutes, but their experiment was partially successful. Skeptics believed that such experiments would lead to real-life Jurassic Park scenarios, and scientists might just start cloning and resurrecting prehistoric mammals. Senior scientist David Wilt from the Smithsonian's National Zoological Park in Washington, D.C., dismissed all such assumptions and considered them impossible. That's all for today. Which one of these experiments did you find to be the most disturbing of all? Let us know your thoughts in the comments below.